The technology being showcased today is Active Virosome and eSame technology platform, versatile platforms for animal vaccine development. The lead inventor is Dr. Vishwas Joshi and the organization is Seagull Biosolutions Pvt Ltd. India's veterinary vaccination program heavily is dominated by conventional vaccine types like live attenuated vaccines and inactivated or killed vaccines, and government programs also reinforce this pattern. Worldwide, MNCs are moving towards new vaccine technologies, and the transition is motivated by a need for faster, safer, and more effective vaccines, especially in response to emerging disease outbreaks. In India, the animal vaccine market size was estimated to be $575 million in 2024, and the major Indian players are Indian Immunologicals, Intas, Zydus, Venky's, Bipcol, Aliviria, etc. And there are various factors driving the growth like government initiatives, wide prevalence, and other factors. What is the gap in India's vaccine ecosystem? Advanced technologies are not easily adoptable. They require high capex and specialized infrastructure and involve complex development and manufacturing processes. That is dependent on global players for advanced solutions, and this leads to missed opportunities for Indian manufacturers. Seagull has two next generation vaccine development platforms offering easy adoptability, affordability, and quick turnaround time for production. These are the Active Virosome technology and the eSame technology for reverse genetics of viruses. So the first technology is the Active Virosome technology. This is useful for producing non-replicated derivatives of measles and other animal virus pathogens including NDV, CDV, rinderpest, PPRV. It mimics an attenuated phenotype. They immunize by themselves and cause limited infection and express viral proteins without causing disease. So they serve as excellent vectors for developing other vaccines. So these active virosome agents produce virus-like particles, protein antigens, RNA viruses in situ, and they can be useful as simple, easy to manufacture, cost-effective, and less capex intensive. The use case for this can be foot and mouth disease virus. Current practice is it is an inactivated vaccine and multivalent. The problems faced are inadequacy of inactivation leads to disease outbreaks, and individual cultivation of different serotypes and their inactivation increases the cost and efforts of production. So the advantages are that by the active virosome technology, it will be easy and hazard-free production, minimal downstream processing, not requiring virus inactivation. Agent is mimicking the attenuated phenotype and easy to formulate into multivalent vaccines. So Seagull can offer to help industrial partners to develop these AV vaccines. The other technology is the eSame technology, that is the episomal self-amplifying mammalian self-expression technology for reverse genetics of viruses. So the virus seeds are the paper sequence and the technology for producing viruses from cDNA. This is an in situ production and it is useful for seed stocks of measles virus, Newcastle's disease, rinderpest, canine distemper virus, Pestide pestis, etc. Useful for producing, like I said, measles virus strain seed. For it has been used by a leading Indian vaccine company, and they have experience for similar development of paper sequence to vaccine seed for other viruses as well. The use case for this would be a PPRV vaccine. The current practice is live attenuated virus vaccines are used, and other strains are based on geographical requirements. So the problems faced are differentiating between the vaccinated and the exposed sheep. So using this technology, attenuated marked PPRV agent which is rescued using reverse genetics, the virus can be recovered from published sequence of attenuated viruses as per the client requirement and artificially marked with appropriate markers for differentiating between the vaccinated and the exposed sheep. So again, they are offering attenuated and marked virus strains for use in vaccine development for clients. Chicken anemia virus vaccine, this is also a use case where it can be used. So current practice is live attenuated virus vaccine is used, and other vaccines under development are subunit virus vectored vaccines. But the problem with the new format vaccines are that they have short-lived immunity and late administration. So what can be done here is using the gene bank sequence, they can express the plasmid and make the vaccine seed stock. They offer to work with industry partners to produce seed stock for this vaccine, and they have experience with producing cDNA for reverse genetics of MVSS, which will help. They don't have the technology, but developing the technology is relatively easy for them because of their experience. What is the value proposition? This new strategy for building vaccines as new serotypes will emerge. The key value proposition compared to attenuated or inactivated viruses is the ability to respond quickly as new serotypes emerge, no risk of poor inactivation, and cost effectiveness in production due to reduced need for production, purification or mixing steps. This will be achieved using the effectiveness which will be comparable to attenuated vaccines. What is the status? They have patents around both the technologies, and for active virosome vaccines, they have worked with companies for chikungunya, Zika and Ebola vaccine agents with Active Virosome UK, and they had a technology collaboration and IP partnership. For technology licensing for vaccine use for influenza, they have worked with Sagitta Biotech Belgium, and they have R&D service contract and licensing agreement. For cDNA to vaccine virus seed stock generation, they have worked with AVT with the client project, and this can also help in reduce import substitution, enable cost reduction, and broaden the access. The company's snapshot, Seagull Biosolutions Pvt Ltd has two proprietary technology platforms, like I said, eSame and AVT, and they have various commercialized products like measles virus seed stock. They have developed for clients vaccines, and they have undergoing clinical trials. The active virosome antigens are being used as clients as reagents for drug discovery applications, and they also have technology which has been licensed to Sagitta Biotech Belgium and presented by Sagitta to BARDA US. The team consists of Dr. Vishwas Joshi, who is the CSO CTO. He is a molecular virologist, and he is the founder and MD of Seagull with more than 30 years of drug discovery R&D. Mukshata Joshi, who is handling the intellectual property. Mukesh Meshram, who is handling the bioprocess and production, and Abhay Patki, who is the clinical development specialist. The ask is they have developed expertise in reverse genetics of viruses and also the technology for producing innovative viral vaccines. So they offer their R&D services to interested parties, and they are also open for licensing. They are seeking to tap those industry partners who are interested in sponsoring further technology development and vaccines, investors who would like to do strategic investments for vaccines or for the platform exploitation, and industries interested in tapping their capabilities as an R&D expert or consultant. For more details, you can reach out to me at these connects.